Hello, my name is Ilham. I'm from Biology Department of Sunan Kalijaga Islamic State University, Yogyakarta. And today, I'm going to work on my experiment. It's about germination media, and light. And it will be so fun and exciting. And so guys, let's go, follow me. Okay, here we at my lab, and welcome guys, this is my lab. It's a botanical ecology laboratory, and I'm going to do my work in experiment here, and you must know that I will carry you in my project in a series of steps that have been pre-arranged and carefully prepared. So, my project title is the effect of light duration and seedling media on germination of monkey. And the objective is to determine whether light exposure and seedling media affect the germination of the monkey. In my experiment, I have two factors. The first factor or light duration. The second factor is germination media, and in a light duration I have three variations. First variation is no light or 0 hours lighting. Second is half daylight or 12 hours lighting, and third is full daylight or 20 hours lighting. And for seedling media, I also have three variations. The first variation is I'm using sand and topsoil that I collected from the topsoil around the laboratory. And third factor is cocoa peat. And you can see the detail here. Okay, the first step, I'm making label for each variation of germination media, and this is for cocoa peat, this is for soil, and this one for sand. And make it stick on this, and I put this like that. And 100 gram for cocoa peat, 150 gram for soil, 150 gram for sand. Next, set up the electricity like this, and making sure that it's work properly. Okay, put the electricity inside the box until like this. First, put the bad quality of the seed that look like this here, and the good quality like this is here. To make sure this is viable or not, soak this on the water. Okay, select the non-viable seed here and the viable seed here. And the viable seed is submarket. I wanna plant this seed here, but before we do that, I wanna spray the media to make this humid media. Plant the mung bean in the media, and here I wanna plant 10 seeds of mung bean on each media. And after I plant the seed here and put the every cup on the box like this. Okay, and cover the plant with the cardboard and making label for every cardboard like this, like I do it before. Is for without like, is for half dialike, and it's for full dialike. Okay, and every germination seed that I plant here will be measured for the environmental parameters and qualitative parameters and quantitative parameters at 7 p.m. and I'm also turn off the lamp for the 12 hours light in 7 a.m. and I turn off again at 7 p.m.
After collecting the data, I finally found that the result of this research. But before we dive in, let's make sure we are on the same page about some morphological terms, just like the one shown in the image. And here you can see there is radicle, there is hypocotyl, and there is pulmula. Okay, we have the environmental parameters. That is so important because we need this to make a consideration about interpreting data, and we have the same temperature and same air humidity, and we have different light intensity because we have different treatment on light intensity and light duration. And first parameter that I measured is germination rate, and as you can see here on the table on the slide, on the chart on the slide, that the distribution looks so pretty, even pretty. As you can see here, this suggests that the light did not have a strong effect on germination rate. Instead, based on the literature, the most influential factor was water content in the growing media and the nutrient. And the second parameter is the radicle length. And based on the graph, it's clear that the best condition for radicle growth was on 12 hours of light in soil media. And as we know that soil contains lots of nutrient and has high porosity, which helps retain water and triggers chemotrophism and encouraging the root to grow. The third parameter is hypocotyl length. As you can see in the graph, that hypocotyl grow longest in soil media with 12 hours of light. And hypocotyl is not appears in the first day because in the first day hypocotyl is in the process of the development, and they appears in the day 2 in the every variation of the factors. The fourth parameter is seedling weight. The heaviest seedling were found on soil media with 12 hours light exposure, and as we know that soil is the most nutrient-rich medium, and 12 hours of light seems to be the optimal condition, because in the 12 hours of light treatment allows the auxin to be produced in a balanced amount, and this how in the 12 hours and in the soil media have the heaviest seedling height. And the fifth parameter is pulmule color. We did not include the media data here, just the 3 images below as you can see here, because all media under the same light treatment have the similar pulmule color, so we skip it for slight efficiency, and that is the reason why I just presenting the 3 images. This is the 3 images that representative pulmule color, and we can see here that in the low level of lighting there is have a pale pulmule because the chlorophyll production was limited, and in the high level of the lighting we have strongly pigment of the pulmule. And the conclusion of this research is the light and growing media did not significantly affect germination rate. Soil media with 12 hours light exposure give the best result for radical length, pulmule length, and seedling weight. And short light exposure led to pale leaves, with longer exposure triggered green pigment production.
Okay, this is my experiment and it's so fun. And okay, guys, thank you for watching and see you on